Okay, I think we'll get started. Good afternoon, everyone. I hope you're all doing well. I have a couple of housekeeping announcements to get going before we start, and that is to please remember to post any of your questions in the Q&A tab, not the chat tab, and I'll be able to get to them and ask your questions to our speaker. And also remember these talks are recorded so that you can see them at the website listed on the screen that you see in front of you. So with that, it is my distinct honor and pleasure to introduce not only a great colleague and collaborator, but a friend, Dr. Shom Gol, who is coming to us from Melbourne, Australia. So I owe him at least a coffee for being here at 3 a.m. His time, which we said is neither early nor late. It's a time everyone should be sleeping. But Dr. Gol is no stranger to the Farber. He did his internship and training here, and prior to that, he did his PhD with Rakesh Jain here in Boston. And he received his medical training at University of Adelaide, and again his PhD here in Boston with Rakesh Jain. And he's now a group leader and medical oncologist at the Peter MacCallum Cancer Center. Dr. Gol has done amazing work, really getting at bedside to bench to bedside work that has had real clinical impact, making beautiful discoveries of mechanism of action of breast cancer progression, mechanism of action of various drug treatments, particularly CDK46 inhibitors, which have been a tremendous success in the breast cancer world. And with that, I will let Dr. Gol tell you more about what he's doing these days. So thank you so much, Dr. Gol, for joining us, especially at 3 a.m.
Thank you so much, Sandy, for that kind introduction. It is early here, or late, depending on how you live your life, but I have to say it's absolutely a pleasure to be here today, and it's an honor for me to be invited to speak at this seminar series. And something that has special meaning for me, I think, as Sandy mentioned, I spent many years in Boston. It was about 10 years of my life that I worked at DFHCC centers. First my PhD at the Mass General, and then 7 years at the Dana Farber, where I worked as a physician scientist in the breast group, doing both clinical work and laboratory work with Jean Zhao. So I have very fond memories of Boston, and in fact, I'm still working very closely with many people in Boston. So before I get started, these are my disclosures that are relevant to this talk, and all of them relate to work about CDK46 inhibitors in cancer, which, as Sandy mentioned, is my main area of expertise, and that's the topic which I'll be focusing on today. So this is where I work now. This is the Peter MacCallum Cancer Center in Melbourne, Australia. I opened my lab here about 2 years ago. Our group is very much focused on cell cycle biology in cancer. And as Sandy mentioned, I also maintain a clinical practice. I see breast cancer patients about 1 day a week. And in addition to doing lab work, I'm also involved in designing and running clinical trials, typically those that are directly related to the things that we study in the lab. And I wanted to take a second here to mention what probably many people don't know, which is that the Peter MacCallum and the Dana Farber Cancer Institute are actually co-signatories to a memorandum of understanding that is designed specifically to foster more and more collaborations between the two institutes, both on the clinical front and research. And I'm actually the manager of that relationship, that collaborative relationship from the Melbourne side. And so I'd really like to take this moment to encourage any of you who have an interest in collaborating with any of our investigators at the Peter Mac, or for that matter, anyone who's interested, for example, in doing a postdoc with us at the Peter Mac, to please get in touch with me. And I'm sure you'll find you're welcomed with open arms. So this is an outline of what I'm going to talk about today. As I mentioned, I work on the CDK46 pathway in cancer, so I'm just going to start with a very brief overview of that pathway. As most of you would know, CDK4 and 6 are really implicated as important regulators of cell cycle transition, and in particular the transition from the G1 phase of the cell cycle to the S phase. And I'm really just including this figure here as a brief orientation for students, for example, who might be listening, who don't necessarily work in this field. Very briefly, CDK4 and 6 enzymes are activated when they're bound with to their partner cyclins, which are the D-type cyclin protein, cyclin D1, 2, or 3. And this hollow enzymatic complex phosphorylates a tumor suppressor protein known as RB. When RB is hypophosphorylated and active, it's bound to these E2F transcription factors and it represses their transcriptional activity. But once it's phosphorylated by CDK4 and 6, it undergoes a conformational change, which leads it to dissociate from the E2F factors. These factors are then de-repressed and are able to drive the expression of a catalog of genes that promote cellular transition into S phase. And when it comes to cancer, and I think breast cancer specifically, we've known for some time now that the CDK4/6 pathway is very important in many breast cancers. And I think a lot of that knowledge really stems from these 3 seminal papers coming from Peter Sosinski's lab, which collectively showed us at a genetic level how important cyclin D1 and CDK4 particularly are in driving the initiation and the maintained growth of carcinomas that are arising from the luminal mammary epithelium. So given all the knowledge that's come from this beautiful work, we've known for some time that pharmacologic inhibition of CDK4 or CDKs4 and 6 would potentially be a very attractive strategy for luminal breast cancer, and perhaps also for several other cancer types. But what I think is very clear is that we couldn't have imagined the potential that these drugs really held or how they might perform until they actually arrived. Now we have, as of now, we have 3 potent selective CDK4/6 inhibitors that are FDA approved for the treatment of breast cancer. And I'm showing those 3 drugs to you here. And I think in the last 5 years, it's very fair to say that these agents have transformed clinical practice in breast oncology. But in addition, I think they've served as very valuable tools in the lab to help us understand a lot more about the CDK4/6 pathway than we've ever understood before, just simply because they are a very nice chemical means to rapidly and precisely turn off the pathway in a variety of model systems. So what do CDK4/6 inhibitors do in breast cancer cells? Well, as you would expect, given the mechanism of action that I described, these agents do induce cancer cell cycle arrest, both in vitro and in vivo, and we typically see cancer cells piling up in the G1 phase of the cell cycle. With more prolonged treatment, maybe several days, for example, in vitro, the drugs will also induce a phenotype that resembles cellular senescence. So cells that become flattened, enlarged, typically show markers of senescence, such as an increase in beta-galactosidase activity. And this is perhaps not that surprising because these drugs work, as I mentioned, by activating the RB tumor suppressor. And we know that RB is a major mediator of the senescence program in cells. So G1 arrest we see, senescence we see. What we're not sure that we see at the moment is actual cell death from CDK4/6 inhibitors. And in fact, the vast majority of data that's out there at the moment would suggest that these agents don't very efficiently induce tumor cell apoptosis directly in luminal breast cancer cells. And I think this is somewhat consistent with the more broad notion that senescence, if anything, is an anti-apoptotic state. And I'll come back to this later in my talk. So this is how we use CDK4/6 inhibitors at the moment. We are using them to treat patients with metastatic hormone receptor positive breast cancer in conjunction with hormonal therapy. And as you can see from outcome curves like this, they have been true game changers in our clinical practice. But I have to say, in my opinion, results like this are really just the tip of the iceberg in terms of what we can achieve with these drugs. We're now beginning to see signs of efficacy for CDK4/6 inhibitors in other breast cancer subtypes and also for other cancer types. And I believe that at this point, it's really imperative that those of us who work in this field have to advocate strongly for the development of these drugs on multiple arenas wherever we think it makes sense. So my lab really has two goals when we study CDK4/6 inhibitors in breast cancer, two very broad goals. Firstly, we are interested in identifying how we can make CDK4/6 inhibitors work better. And by that, what I really mean is identify new therapeutic combinations, things that we can add to CDK4/6 inhibitors that will enhance their efficacy. And we're equally interested in understanding why it is that these drugs stop working over time. In other words, mechanisms of acquired resistance. And so to my thinking, if you ever want to understand how to make anything work better, or for that matter, to understand why anything stops working, you first really need to understand how it works. And that's why our studies in the last few years, as Sandy mentioned, have been so focused on exploring the mechanisms by which these drugs exert their effects in cancer. And this has taken us down a number of different paths. And at the moment, my lab is engaged in studies of basic cell cycle biology. We're also very interested in studying the RB protein itself, and of course, focused on a lot of studies using these drugs. And more broadly, because of everything I've explained, we've developed a strong interest in therapy-induced senescence in cancer as well. So given that I'm so interested in understanding how these drugs work, I thought I would include a slide here which presents a framework that I use when I'm thinking about CDK4/6 inhibitors and how they exert their mechanisms of activity in cancer. So as you can see, and as I've mentioned, hypophosphorylated RB, which is what these drugs essentially achieve, binds to and represses the E2F factors here and causes G1 arrest. But as I also mentioned, these drugs can induce a senescent-like state in cancer cells. And it's quite possible, although not always proven, that a number of the features that we think about in classical senescence, which is typically studied in benign fibroblasts, may also be seen in cancer cells which are treated with CDK4/6 inhibitors. In addition, it's really important also to realize that this catalog of S-phase genes that is downregulated by these drugs includes a number of really important epigenetic and metabolic enzymes. And although these are enzymes that are primarily required in S phase, which is why they are E2F targets, they can also have roles in G1, and their depletion in the face of sustained G1 arrest that happens with these drugs can also have significant consequences for a cancer cell. And another, I think, perhaps less appreciated fact is that hypophosphorylated RB does a lot more than just repressing E2F factors. And I'm going to show you an example of this later. But suffice it to say at this point that any other properties of hypophosphorylated RB will also be brought out by these drugs. Another somewhat inconvenient truth is that CDK4/6 inhibitors have many non-RB substrates, which are also impacted by these agents. And so we must keep this in mind when we're thinking about the cellular effects of CDK4/6 inhibitors in tumor cells. And finally, we must also keep in mind the fact that when a patient receives these drugs, other cell types within a cancer, immune cells, fibroblasts, endothelial cells, etc., are also exposed to the drug. And changes in their biology in direct response to the drug could also have profound implications for the ultimate drug response that we see. So with all of this in mind and this complex framework about thinking about how these drugs work, what I'd like to do now is describe to you throughout the course of this presentation, 3 studies that we have performed, which show how some of these lesser appreciated effects of CDK4/6 inhibitors can actually be very important and also clinically relevant. And I think it's fair to say that these 3 projects that I'll discuss have really changed the way I think about these drugs. The first 2 studies are slightly older, and some of you may be familiar with these, so I'm only going to describe them quite briefly. But the reason I wanted to share them with you today is because I'm very excited to provide some updates on how we have taken the results of these projects to move our findings towards the clinic. And we are really getting to a point now where we're beginning to see real clinical impact that's directly stemmed from this preclinical work. And then I'll spend the rest of my time discussing a more recent study that our group has published showing how CDK4/6 inhibitors influence the cancer cell epigenome. So to the first story, I really want to go back to this study here. This This story stems from the paper that I published whilst I was a postdoc in Jean Zhao's lab in Boston. And in this work, we identified that CDK46 activity, CDK46 enzyme activity, can actually drive resistance to inhibitors of other growth factor receptor tyrosine kinases in cancer cells. So in this case, we focused on HER2 inhibitors in the context of HER2-positive breast cancer, but the broader message that came out of this work really applies to several different receptor tyrosine kinase inhibitors that drive proliferation across a number of different cancer types. The project actually didn't begin as a cell cycle study. When I started this work as a postdoc, we were really focused on trying to understand the mechanisms of therapeutic resistance to HER2 pathway blockade in HER2-positive breast cancer. And we initiated that work using this genetically engineered mouse model, which is characterized in the paper. In this mouse model, doxycycline is used to induce the expression of an oncogene that's wild-type human HER2 in this case, specifically in the adult mammary gland. And this ultimately leads to the development of adenocarcinomas in these mice. And importantly, once these tumors are developed, we can mimic HER2 pathway blockade genetically by withdrawing doxycycline from the mouse's diet. This simply switches off HER2 expression and leads to rapid and complete tumor regression. What's really interesting in this model is that if you continue to observe these mice, the vast majority of them will experience a spontaneous tumor recurrence in the mammary gland several months later. And it turns out that with lots of careful searching, one can actually identify these pockets of residual tumor cells that survived the original HER2 withdrawal and which ultimately go on to give rise to the recurrence months later. And it was our studies of these cells that were able to survive complete HER2 pathway blockade that led us to ultimately conclude that the CDK46 pathway mediates resistance to HER2 pathway blockade. And ultimately what this did was it led us through a whole series of studies where we eventually showed that if you have HER2 positive breast cancer cell lines, for example, as I'm showing you here, which are resistant to conventional anti-HER2 therapy. So here I'm looking at the tyrosine kinase inhibitor, lapatinib. The addition of a CDK46 inhibitor, in this case, abemaciclib, can overcome that resistance. And these plots demonstrate really potent synergy when one combines a CDK46 inhibitor with a HER2 kinase inhibitor in these cells. From a mechanistic standpoint, we found that the combination of HER2 pathway blockade and CDK46 pathway blockade led to sort of maximal inhibition of tumor cell proliferation. That was the main effect that we saw. And specifically what we observed was that although CDK46 inhibition, so here I'm looking at abemaciclib, the third lane of this Western, can completely shut down phosphorylation of RB, at least at this residue, it was only when we combined these two drugs together that we also saw marked suppression of the readouts of mTOR activity too. So here I'm looking at phospho S6 kinase, for example, and phospho S6. And as many of you will know, mTORC1 is also another important driver of the G1 to S transition. And we believe that it is this combined inhibition of RB phosphorylation and S6 phosphorylation or mTOR activity that ultimately explains the synergistic arrest in G1 that you see when you combine these two agents. We then went on to try and explore why this was the case. And what we really observed was that in cells, so here I'm showing you a classical cancer cell that is driven by any number of different upstream receptor tyrosine kinases. In cells that are resistant to receptor tyrosine kinase blockade, we were seeing that inhibition of the receptor tyrosine kinase led to suppression of mediators immediately downstream, for example, PI3 kinase activity or measured as phospho-AKT as an example. However, we were not seeing marked suppression of mTORC1 downstream in these resistant cells. And we observed that this was because in several cell lines, it was actually persistent CDK46 activity that was sustaining mTORC1 activity as well. And it was only when we combined inhibition of CDK46 and the upstream RTK that we were able to shut down both phosphorylation of RB and mTOR activity, and hence saw potent G1 arrest. And really importantly, we showed that this direct stimulation of mTOR by CDK46 was being mediated through this tumor suppressor TSC2. We demonstrated here that both cyclin D and CDK4 directly bind to TSC2, which is a negative regulator of mTOR. And we also showed that CDK46 inhibitors inhibit the phosphorylation of TSC2 at certain residues. And this suggested to us that TSC2 might even be a CDK4 substrate, although we didn't specifically show that in this work. It turns out that a very similar concept had actually been explored about 15 years beforehand in this paper from the University of North Carolina, albeit in somewhat less detail. But more importantly, I think since we published our work, Aurelio Telemann's lab at the DKFZ has really elegantly explored this connection between CDK4/6 and TSC2 in much more detail, both in Drosophila and in human cells, and really now has actually confirmed that TSC2 is indeed a bona fide CDK4 substrate. So what do we do with this information and this observed synergy we've made in the lab? Well, over the last few years, we've been working really hard to design and implement trials in the clinic where we can test these concepts out. So here are the results from the first of these trials. This is a trial for patients with metastatic HER2-positive breast cancer, led by Dr. Sarah Tillany at the Dana Farber. And these are patients whose cancers have developed resistance to our standard HER2 inhibitors. And what we found in this study was that using a CDK4/6 inhibitor, abemaciclib, with anti-HER2 therapy outperformed the standard of care treatment, which would be to use chemotherapy in conjunction with an anti-HER2 drug. And we think these results look really promising. And this concept is now being extended in a number of other clinical trials in HER2-positive breast cancer, a couple of which are being designed with the goal of FDA registration. And so if these studies are positive, and we're optimistic about them, they have the potential to bring CDK4/6 inhibitor, HER2 inhibitor combinations into mainstream clinical practice. In addition, there are a number of other trials that are ongoing that are testing this broad concept of RTK plus CDK4/6 inhibition in other cancer types, based largely on a number of preclinical studies which have now been completed, showing similar effects on S6 phosphorylation with combination treatment. So if we take all these lessons and go back to this framework about CDK4/6 inhibition in cancer, I think this is a classical example of how a non-RB substrate, so in this case TSC2, becomes really important when we think about how the drugs exert their activity and also how we might want to develop them in the clinic. Okay, so that's example one. Now I'm going to switch gears a little bit and tell you about a second project that we've done, and again, show you how that's translating into a clinical development approach as well. So the second example is based on this study, which is work that I also did as a postdoc with Jean and Sandy in Boston, and it's a really fascinating subject to me, and one that still remains a major focus in our lab. And what we really showed in this work was that CDK4/6 inhibitors can actually stimulate anti-tumor immune responses. Certainly, this was not the reason why these drugs were developed, as I explained at the beginning of this talk. And it was also quite unexpected, especially because there were concerns that the opposite would be the case, that CDK4/6 inhibitors would actually impair anti-tumor immunity by inhibiting the CDK6-mediated proliferation of T lymphocytes. This slide just shows you some of the initial experiments that we did in the lab that led us to believe that there might be an immune component to CDK4/6 inhibitor activity. So to run you through these studies very briefly. So again, I'm talking about that same transgenic mouse model that I explained before. It's an immune competent animal. What we found in the lab was that if we treated tumor-bearing mice with a CDK4/6 inhibitor, we used abemaciclib, we did, as expected, see a reduction in tumor cell proliferation, but we also saw tumor regression. And this is actually similar to what we see in a proportion of patients treated with this drug in the clinic. However, if we took these tumors and transplant them orthotopically into the mammary glands of athymic mice, so these are mice without an adaptive immune system, we still see suppression of tumor cell proliferation, but we no longer see regression. We only see growth delay, and this has been consistently seen over numerous experiments. Perhaps even more convincingly, if we take the original immune competent mice and deplete CD8 positive cytotoxic T cells from them using a neutralizing antibody, we find that the CDK46 inhibitor can arrest tumor growth, but no longer causes tumor regression. So it's this body of experiments that collectively led us to think that there might be an immune mechanism at play. But why would that be the case? Why would a CDK46 inhibitor trigger anti-tumor immunity? Well, since we published our work, a number of other groups have shown something similar, and collectively, I think we've all added to the picture, and we're beginning now to understand the complex mechanisms at play here and numerous mechanisms that are at play. So the first component to the mechanisms of how this is happening, we believe, is that CDK46 inhibitors can directly impact tumor cells to enhance their immunogenicity, and particularly by encouraging antigen presentation through MHC class I. And again, this ties back to this notion that CDK46 inhibitors can induce a senescent-like state in cancer cells. So this is data from classical senescence in benign fibroblasts, so nothing to do with cancer here. And what we've known for some time, based on work like this, is that classical senescence is actually known to be a state of global DNA hypomethylation. There are focal regions of hypermethylation, but at a global level, we see hypomethylation, and this is associated with reductions in the levels of the maintenance DNA methyltransferase DNMT1, and also changes in DNMT1 localization within cells. What we found was that when one treats luminal breast cancer cells with a CDK46 inhibitor, you see similar reductions in DNMT1, and at a global level, reductions in total 5-methylcytosine levels in tumor cells as well. And what's really important when we come back to this question of immunogenicity is that we observed that certain hypomethylated regions in these cancer cells after treatment directly overlay repetitive elements in the genome. For example, the endogenous retroviral elements. And the consequent expression of these regions after hypomethylation led to the production of double-stranded RNA, which was measurably increased in tumor cells. And this set off a response known as viral mimicry, or an antiviral response within the cancer cell characterized by interferon production and therefore enhancement of antigen presentation. So this is potentially one mechanism by which these drugs are having this effect. But on the other hand, the effects of these drugs on T cells directly, on immune cells, could also be playing a really important role. So first of all, it appears that although the proliferation of various T cell subsets is somewhat impaired by these drugs, it seems that the proliferation of immunosuppressive regulatory T cells is particularly sensitive to CDK46 inhibitors, such that treatment with these drugs leads to a shift in the Treg to CD8 ratio towards one that favors anti-tumor immunity. And that's been shown by a number of groups now. Secondly, others have subsequently shown that not only can CDK46 inhibitors deplete Treg numbers in tumors, but that they can also directly impair the suppressive function of Tregs. So this is a co-culture assay where Tregs are pre-treated with a CDK46 inhibitor and then found that this pre-treatment impairs their ability to restrain the proliferation of CD8 positive cells. So this is a really interesting phenomenon, but I think it's fair to say that we don't really understand the mechanisms underlying this at the moment. And thirdly, CDK46 inhibitors have also been shown to directly stimulate T cells. And this appears to be because CDK6 in particular can directly phosphorylate members of the NFAT transcription factor family, perhaps NFAT2 or 4. And inhibition of this phosphorylation with a CDK46 inhibitor leads to retention of NFAT in the T cell nucleus and enhanced T cell function. So collectively, we see a body of mechanisms, some in the tumor cells, some in the immune cells, which collectively one might expect could in fact boost anti-tumor immune responses despite an anti-proliferative effect to some extent in T cells. And certainly this phenomenon of the boosting of anti-tumor immune responses is what has been reported in vivo. So this is what we see, for example, in our original mouse model. Mice were treated here with a CDK46 inhibitor for a couple of weeks, and then transcriptomic profiling of the tumors was performed, and we can see upregulation of a variety of gene expression signatures that we might associate with warming up of the tumor immune microenvironment. And more recently, we've seen very similar results in clinical specimens. So this is neoadjuvant studies in hormone receptor positive breast cancer. And we can see that in trials using two different CDK46 inhibitors, the same gene expression signatures appear to be upregulated when one compares post-treatment biopsies to pre-treatment biopsies. So there is clinical evidence that this phenomenon may actually be real. And as you could imagine, the first thing that comes to your mind after that is, well, how can we leverage this to help our patients? And I think obviously one obvious thought is that if you have an agent that's enhancing anti-tumor immunity, perhaps it could be successfully combined with immune checkpoint inhibitors as well. So we looked at this in the animal models. This is just our data using the CT26 colorectal cancer model, actually, showing really marked synergy when one combines a CDK46 inhibitor with an anti-PD-L1 antibody. And actually, the data is being reproduced. This is all from different labs, all using the same mouse model, but all showing similar effects, this synergy between CDK46 inhibition and immune checkpoint inhibition. So based on these sorts of results, a number of clinical trials have now opened testing CDK46 inhibitor plus immune checkpoint inhibitor combinations. And we're seeing these trials not only in breast cancer, but in melanoma, in lung cancer, and in other settings as well. And early data is actually suggesting that there are some very nice responses being observed in treatment refractory patients. The potential issue that we're running into in these studies, unfortunately, is one of toxicity. And it's somewhat ironic, but in the early results that we've seen, it seems that the problems we're running into might be actually an excess of immune-mediated toxicity, so hepatitis, pneumonitis, etc., which may, although we're not sure, on the one hand, be proof of principle that in fact these two drugs together do boost sort of host immunity more generally, but it may also be a problem that limits the development of these combinations in the clinic. So how could we take this concept further then? If we're a little concerned about just routinely combining these drugs with immune checkpoint inhibitors. Well, there is another CDK46 inhibitor that I'll mention here called trilaciclib, which is being developed at the moment specifically with these ideas, immune mechanisms in mind. Unlike other CDK46 inhibitors, the manufacturers of trilaciclib, that's a company known as G1 Therapeutics, actually originally founded by Ned Sharpless before he was at the NCI. That company has really focused less on how this drug affects the proliferation of tumor cells and more on how it impacts hematopoietic cells. And in fact, the drug was initially developed with the idea that it would induce G1 arrest in hematopoietic stem and progenitor cells in the bone marrow, with the goal of preventing chemotherapy-related toxicity in the bone marrow. And in fact, the drug was just granted FDA approval for this exact purpose in the context of certain subtypes of lung cancer. More recently, G1 Therapeutics has focused their attention more on the immune stimulatory properties of trilaciclib. And the company's done a lot of work to understand these effects. And here's just a couple of preclinical experiments, for example, showing how the addition of trilaciclib, these are the orange lines in this colorectal cancer model, markedly enhances the anti-tumor effects of a chemotherapy plus immune checkpoint inhibitor combination. What's been really exciting to me are the striking results of this randomized phase 2 clinical trial here in triple-negative breast cancer. So in this trial, patients were treated with metastatic triple-negative breast cancer, were treated with chemotherapy, which is the standard of care, with or without the addition of trilaciclib administered prior to each chemo infusion. And in this study, actually, trilaciclib was given with that original goal that I mentioned, which was to reduce chemotherapy-related toxicity in the bone marrow. Turns out that that actually didn't happen. That outcome was not met in the trial. But what was seen was this marked prolongation of overall survival in the two treatment groups that received trilaciclib therapy. And as you might imagine, this is a very aggressive disease, triple-negative breast cancer with relatively few treatment options. This result has garnered a lot of attention. At this stage, I think the mechanisms behind this improvement in overall survival are not entirely clear. However, this immune stimulation hypothesis that I have described to you is probably at the top of the list. And there is very limited data from the peripheral blood, of patients on this study, suggesting that this immune mechanism might actually be in play here. So here we can see, looking at cytotoxic T cells in the peripheral blood of patients on this study, that those cells taken from trilaciclib treated patients show an increased capacity to make interferon gamma upon re-stimulation ex vivo. And I understand all the caveats that come with an assay like this, and that it's not a perfect proof of concept at all, but nonetheless, it's extremely intriguing. So given all of this and this new direction that this drug is taking, I've been working really closely with G1 Therapeutics to put together a larger definitive clinical trial, which will help firstly really establish whether this survival benefit is real, and if so, why it's happening. And so we've designed this new clinical trial called the PRESERVE-2 trial. This is a global randomized phase 3 study, which will be opening very soon for patients with metastatic triple-negative breast cancer. And basically, this is the design of the study. So patients will either receive chemotherapy, that's GC, gemcitabine and carboplatin, with or without intermittent administration of trilaciclib. And the study is much larger than the randomized phase 2 I showed you. It's 250 patients, and it's designed to definitively answer the question of whether trilaciclib does in fact improve overall survival in this population. And what's really exciting to me, and which is quite unusual in a randomized phase 3 trial, is that we've built in an 80-patient paired biopsy sub-study to the trial. This is a rarity in randomized phase 3 trials, which will allow us to address the question of why the benefits that we might be seeing are actually happening in patients and resolve some of these questions about mechanism. So to take a step back then, putting all that immune data together, and going back to our framework of thinking about how these drugs exert their effects, I think it's another really good example of how some of the less classical effects of CDK46 inhibitors could actually be really important. So I talked about how the inhibition of DNA methyltransferase 1, which is an E2F target gene, may have a role to play in antigen presentation in tumor cells. We talked about how these drugs can also impact other cell types, in this context, T cells, and other groups, as I showed you, have also shown how non-RB substrates, in this case, the NFAT factors in T cells, may also be important in explaining mechanisms of activity. So for the final section of this talk, I'd like to focus now on some more recent work that we've published earlier this year, looking at how CDK46 inhibitors remodel the cancer cell epigenome. And this is a study that was led by three very talented women, April Watt, who's a PhD student in my lab at the Peter Mac, Paloma Sayas, who's a senior scientist at the Dana Farber's Center for Functional Cancer Epigenetics, and Molly DeCristo, who's now a medical student at the University of North Carolina. And we did this work very heavily in collaboration with the CFC at Dana Farber, working with Henry Long and Miles Brown as well. The reason we were interested in understanding how CDK46 inhibitors might impact the cancer cell epigenome goes back to this observation that I mentioned at the start, that these drugs can induce a phenotype that resembles senescence in cancer cells. And we were aware that in recent years, several groups had spent time characterizing the epigenome of senescent cells. But as is often done in senescence, the studies were done in benign fibroblasts, not in cancer cells. And what's become really clear in recent years is that epigenetic remodeling in senescent cells in classical senescence is profound. And it's not only to do with the development of heterochromatic foci and repression of gene expression that we typically think about in senescence, but as these studies have shown, senescence is also characterized by the activation of a wide range of new distal regulatory elements, enhancers within cells. And so the question we were asking is, does CDK46 inhibitor induced senescence in a cancer cell look anything like this? These are H3K27 acetylation ChIP-seq plots showing these newly active enhancers in senescent cells. Do we see similar patterns of enhancer activation in cancer cells? And if we do, what does it look like? And what are the implications for the tumor biology and how it's responding to the drug? So the short answer, to summarize what we found in this study, is that CDK46 inhibitor induced senescence does, at least in part, phenocopy some of the epigenetic changes that are seen in classical senescence in benign fibroblasts. So what do I mean by that? Well, what we've done in this work is originally taken a number of luminal breast cancer cell lines, treated them with CDK46 inhibitors until a senescent morphology is seen, so a few days of treatment in vitro at least, and then assessed their epigenome using a variety of techniques, predominantly ATAC-seq to look at changes in chromatin accessibility, and also ChIP-seq for a variety of enhancer marks. And as I say, much of this work was done in collaboration with the CFCE. And here's an example of what we found looking at MCF7 breast cancer cells. As you can see, CDK46 inhibition with abemaciclib here induced gains in chromatin accessibility across thousands of different genomic regions in these cells. And these regions were almost exclusively in distal intergenic regions or in introns, and were decorated with increases in H3K27 acetylation after treatment, suggesting to us that they may well be newly activated enhancers. What's really interesting is that we see very similar changes in vivo. So here we performed H3K27 acetylation ChIP-seq on paired biopsies taken from a patient, taken before, this is in orange on the left, and after a course of CDK46 inhibitor treatment. This is a patient that's enrolled on a clinical trial being led by Dr. Otto Metzger at the Dana Farber called the PELOPS trial. And what we found on this ChIP-seq experiment was that many of the same genomic regions which were showing enhancer activation in vitro, so in blue is what we're seeing in cells, were also showing gains in H3K27 acetylation in this clinical analysis as well. The next thing we went on to look at is whether this was an RB-dependent sort of on-target effect of the drugs, and we found that it was. So what we're seeing here is that when we take these MCF7 cells and knock down expression of RB1, we no longer see these increases in chromatin accessibility after a week of abemaciclib treatment. So what we were really interested in at this point is asking the question, well, what do these new enhancers do? What biological processes might they regulate? Is there any method to this madness, or is this just a random collection of regulatory elements? And so we've looked at this in a lot of detail by performing ontologic analysis of these of the genomic regions that are covered by these new enhancers, correlating that with extensive RNA-seq analyses. And to summarize, we found a number of discrete biological processes that we believe are regulated by these new enhancers in breast cancer cells that have been treated with CDK46 inhibitors. And I don't have time to go over everything in great detail, and some of these biological phenomena remain the subject of ongoing studies that we're doing to confirm exactly what's going on. But what I will do is show you a couple of key examples where I think we're fairly confident about the biology. So once we'd identified these new enhancers, one of the first things we did was perform, as I said, ontologic analysis of the new enhancer regions. And what we found was that many of these new enhancers were situated very close to genes that regulate mammary epithelial development, and in particular, regulate the expression of genes that we think of as being expressed in luminal epithelium in the mammary gland, remembering that these are cancer cells that are thought to have originally been derived from the luminal epithelium. And using both ChIP-seq and also high ChIP, this is at the bottom here, high ChIP for H3K27 acetylation, we see marked activation of enhancers around many classical luminal genes. This is just an example, as well as upregulated expression of these classical luminal genes in many breast cancer cell lines treated with CDK4/6 inhibitors. And remarkably, this effect can even be appreciated, the ultimate phenotype can be appreciated when one looks at these tumors under the microscope. So here we're seeing in a mouse model, the same mouse model that I've been talking about, that prolonged CDK4/6 inhibitor treatment increases what we call tubule formation histologically in the tumor, which is consistent with a better differentiated state. And going back to the patient that I described earlier, whose tissue we were able to analyze, we see exactly the same thing. So here we see activation of enhancers over luminal cytokeratin genes, so keratin 818, both in a cell line and in exactly the same regions in this patient's tissue after CDK4/6 inhibitor treatment, and also evidence of, on the right here, increased tubule formation in this cancer after CDK4/6 inhibitor treatment. And to me, this body of observations is interesting, not only because it explains how these drugs might impact differentiation in luminal breast cancer, but also because I think the data provides an epigenetic link between the inhibition of cellular proliferation and the enhancement of differentiation. So it's often discussed that that these two phenomena are inversely associated, but here we're beginning to see an epigenetic connection between the two, and specifically beginning to understand the role that RB might play in governing both proliferation and differentiation at the epigenetic level. A second biological process that we found that these enhancers can also regulate is apoptotic evasion. And as you might remember at the beginning of this talk, I mentioned that senescence has long been considered to be an anti-apoptotic state outside of cancer. And what we have found is that when breast cancer cells are put into a senescent-like state with a CDK4/6 inhibitor, they also sort of become primed away from apoptosis and do become relatively resistant to classical apoptotic insults. So here what we're seeing, for example, is that pre-treatment of, I think this is MCF7 breast cancer cells with the CDK4/6 inhibitor abemaciclib, mitigates the apoptotic response that we get as a consequence of treatment with staurosporin. So here's staurosporin causing marked apoptosis in these tumor cells, and we just don't see that in the same tumor cells that have been pre-treated with a CDK4/6 inhibitor. And interestingly, this is somewhat reflected in the results of a recent clinical trial showing that the addition of a CDK4/6 inhibitor, palbociclib, to endocrine therapy in breast cancer actually suppresses rather than increases markers of apoptosis, here looking at cleaved PARP. So when we saw this, we were very fortunate to be able to work with Tony Latai's group, and we learned that this phenomenon, this apoptotic resistance, if you will, was resulting directly from because these cells were primed away from apoptosis at the level of the intrinsic apoptotic pathway. Working with his group to perform dynamic BH3 profiling, and specifically what we found was that this phenomenon relates to the activation with CDK4/6 inhibitors of a super enhancer over the BCL2L1 locus, which encodes for the anti-apoptotic protein BCLXL. Here you can see that happening in cells in the top two rows, and then below in the clinical sample that I've been describing throughout the course of this presentation. And this ultimately leads to increased intratumoral levels of BCLXL protein as well. What was remarkable to us was that when we then sort of tried to translate these findings into in vivo models, we found that CDK4/6 inhibitor pre-treated tumors, so I'm looking at this column here called combination, showed particular sensitivity to BCLXL inhibitors in vivo. So what that suggests to us is that, in fact, this state of senescence that CDK4/6 inhibitors induce is actually associated with increased dependence on BCLXL for tumor cell survival. And once again, to me, this is not only interesting from a breast cancer standpoint, but it also speaks to this more fundamental idea that senescence is an anti-apoptotic state, and it provides an epigenetic mechanism perhaps behind that observation, and connects it as well to the activity of RB. So when you see all these enhancers being activated and these profound changes in tumor cell biology, one question that obviously springs to mind is, well, why is this all happening? And I think mechanistically, where we're up to at the moment is that we believe that the AP1 family of transcription factors is heavily involved in the epigenetic remodeling that we're seeing in these breast cancer cells. And we came to this conclusion by performing an unbiased analysis of the newly activated enhancer regions that we were seeing in both in vitro and in vivo specimens. And having found that these regions were heavily enriched for AP1 binding sites, we then went on to study AP1 biology in these cells in more detail. And the first thing we found, which really struck us, was that CDK46 inhibitor treatment dramatically increases the levels of various AP1 factors in the nucleus of breast cancer cell lines in vitro. And in addition, when we look at the newly activated enhancers here, looking at the ATAC, the K27 acetylation in vitro, we find that many of these show increased occupancy of different AP1 factor family members after CDK46 inhibitor treatment as well. And supporting this idea that the AP1 factors may be involved, we also found that the expression of various genes encoding different AP1 family members was dramatically upregulated in clinical specimens taken from patients treated with a CDK46 inhibitor. So these are samples taken from a neoadjuvant trial. We're looking at about between 25 and 35 patients per time point here, serial biopsies from the same patients. And it's between this cycle 1, day 1 and surgical tissue, this second and third points, that the CDK46 inhibitor has been administered. And what we found was that of the 15,000 genes that are expressed in the microarray analysis of these tumors, these 4 AP1 factors were all among the 35 most upregulated genes in tumors after CDK46 inhibitor treatment. So we were really starting to think that AP1 factors may be involved in this epigenetic remodeling. And what's really interesting to us is that older literature, again, we often find the answers to what we're trying to understand in the older literature, suggests that there may be already a mechanistic explanation for this. Hypophosphorylated RB, which is what these drugs induce, is actually known to be able to bind to certain AP1 factors, notably c-Jun, but also others, and activate their transcriptional activity. And it's well known that AP1, once activated, can drive its own expression in a feed-forward loop. And we believe that it's this process that leads to a dramatic buildup of AP1 factors in these cells over time when they're treated with a CDK46 inhibitor. And it's that then allows the AP1 factors to force changes in enhancer activity. What's really important to mention as well is that if we try to inhibit the effects of AP1, and we chose to use a chemical method, so this is a pharmacologic compound that represses AP1 complexes, we found that we can switch off or reverse some of the transcriptional programs that are induced by CDK46 inhibitors in breast cancer cells. So what you're seeing here, for example, is the expression program associated with luminal differentiation in breast cancer cells being mitigated when this AP1 inhibitor is added to a CDK46 inhibitor. And what further supports our thinking, I believe, in this work is a number of other studies that have all come out quite recently, most notably this work from Oliver Bischoff's lab published in Nature Cell Biology, all of which are starting to converge on this notion that the AP1 factors play a really important role in the biology of cellular senescence. And again, we believe this ties back to what's already been shown in the past about connections between AP1 and hypophosphorylated RB. So taking this third story back to our framework again, I think here we have another impact of these drugs in cancer cells, which I believe is quite profound. This epigenetic remodeling has very broad implications for understanding how the drugs work. And again, relates to a non-classical impact of the drugs, as previously described, an underappreciated effect of hypophosphorylated RB. So that brings me to the end of my talk. That's these three sort of different stories, three different effects of these drugs, all occurring through perhaps mechanisms that one would not typically associate with these drugs. So I hope what I've been able to show you through these three stories is that, first of all, not only can these drugs have unexpected activities, but many times they can be of great clinical relevance. And also just reinforce this idea that now that we have these very selective and potent inhibitors of CDK46, we really have a very powerful toolkit to understand pathway biology in much more detail, and also to begin to interrogate many more questions about cellular senescence. So on my last slide, I'd just like to briefly thank a few people, or a number of people actually, who've worked with me over the years, most notably the members of my lab at the Peter Mac, who are listed here. They've done a stellar job. You know, the lab opened in mid-2019, and shortly thereafter we sort of shut down because of the COVID pandemic, and they've still managed to, keep good spirits and keep their work going despite that, which has been wonderful. To thank everyone at the Dana Farber who I worked with over many years, most notably my mentors, Eric Weiner and Jean Zhao, my clinical colleague, Otto Metzger-Filo, and also members of the CFCE at the Dana Farber, who we've worked with on the epigenetics projects, various other collaborators throughout Australia listed here, and also the various agencies and foundations and companies that have funded this work. And so with that, I'll close up, and I'm very happy to take your questions. Thank you.
Great, thank you, Shom. That was phenomenal. A real tour de force, and really important insights. Thank you for that. I wish you could hear the applause you're getting.
Thank you, Sandy.
A problem with having virtual meetings, but we do have a number of questions, and I'll have to say we do have some time, but if I can't get to all of the questions, Claudia will be sending them to Dr. Gold so that he can answer offline if that's what you prefer. So let's start right in with the questions. This came in earlier in your talk from Natalia Rameka, asking, does the CDK46 immune activation mean that we could potentially use their combination with CPI upon progression in hormone receptor positive breast cancer?
CPI, I'm guessing, means checkpoint inhibition. So I'm guessing so. That would make sense.
Yeah.
Look, it's a really good question. It's certainly something that's already being tested in the clinic. So there's actually a clinical trial that's being run by Dr. Erica Mayer at the Dana Farber, looking at this exact question. Do patients whose hormone receptor positive breast cancers have developed resistance to CDK4/6 inhibitors benefit from the addition of immune checkpoint inhibitors? So we will get an answer to this question in the years ahead. I think one of the concerns I've always had is that when we think about, we've used CDK4/6 inhibitors in the lab to demonstrate these immune effects of the drugs. But in the clinic, these drugs have been very much developed as treatment for ER-positive metastatic breast cancer. One of the issues is that metastatic ER-positive breast cancer is not considered to be a particularly sort of immune-driven disease. And a concern that I've always had is that trying to translate our laboratory results purely to hormone receptor positive breast cancer and say, well, if this combination works in hormone receptor positive breast cancer, it means that it's a true effect is a little bit risky. I think it's really important, as I say, there are trials ongoing, but it's really important that we look at how these CDK4/6 inhibitor IO combinations work in other cancer subtypes where we think we can perhaps leverage anti-tumor immunity more easily.
Right. Thank you. There's a question from Min Wang asking, will a more selective CDK6 inhibitor perhaps be a better partner with immune checkpoint blockade?
Yeah, that's a great question. So again, the thinking at the moment, although there's so much, it's very complex because there's a lot of redundancy, right, between CDKs 4 and 6. And when you inhibit one in certain cell types, the other one can take over, which always makes things complex. But the simplistic thinking is that a lot of the effects we see in T cells are mediated through CDK6. And at least on the surface, a lot of the effects we see in luminal breast cancer cells may be mediated through CDK4. As I explained in my talk, we think that some of the immune effects are happening because of the tumor cell related effects of these drugs. So therefore CDK4 might be important. Many of the effects may be happening through the T cell related effects, so CDK6 would be important. And I think the honest answer is that the relative contribution of the tumor cell effects and T cell effects isn't known. We're actually getting at this right now by making some new transgenic animals where we grow these HER2 positive breast cancers, but simultaneously delete RB. And then we'll be able to look, so in the tumor cells specifically, and then we'll be able to look that if we limit the effect of the drug purely to the immune compartment, to what extent is this immune phenomenon still seen? But I think it's an open question at the moment.
Right. Stephanie Dugan says, "Hi, Shom. Why do you think there's no enhancement of AP1 at day 1? Why does it take 7 days?"
Yeah, that's a great question. And hi, Stephanie. So I think what I showed in the slide there was maybe some Westerns with day 1 and day 7 very quickly. If we look at it in more detail, what we find is that when we put these drugs on luminal breast cancer cells, the expression of various AP1 factor genes, and most notably c-Jun, is upregulated within hours. So within 6 hours. So that's consistent with the mechanism that I hypothesized, that hypophosphorylated RB is binding to AP1 and driving AP1 transcriptional activity. The first time point that we looked at the protein level is at 24 hours. At 24 hours, we see very slight increase in protein. Some factors more than others, but we do believe that this process has already kicked off by then. And then at 7 days, which is the next time point we looked at, everything is much, much more exaggerated for reasons that I explained. So yeah, we do actually, and what we've also done, which I didn't have time to show, is serial ATAC sequencing on these breast cancer cells treated with CDK46 inhibitors at a variety of time points. So 24 hours, 48 hours, all the way out to 7 days when the senescent morphology is really clear. And although these regions of increased chromatin accessibility are most pronounced at later time points, we can actually see early signs that these new enhancers are being activated even within 24 hours. So I really think that it's just a continuous process that starts early but becomes more pronounced as time passes.
Yeah, thank you. So Marcos Malumbres is asking a question. Any indication of specific molecular effects of abema that are different from those of palbo-ribal? Maybe I'll expand upon that. I think, you're finding new, unexpected, on-target pathways and effects of these drugs. I guess, is there a distinction, and that's what perhaps Marcos is asking, with off-target effects, and how important will it be to distinguish those?
So hi, Marcos, first of all, and thanks for that great question. I think there's very little that I can teach Marcos about these drugs. So, just with respect to our lab work specifically, for all of the phenomena that I showed in these studies, we have confirmed that they happen with both abemaciclib and at least one other inhibitor. And we typically have used palbociclib as the second drug. And many other labs who've sort of replicated some of these findings have seen something similar. That said, we do know that these drugs are not all the same. If you go back to chemical structure, we know that ribociclib and palbociclib are much more structurally related than abemaciclib. In vitro kinase assays would suggest that abemaciclib has a broader spectrum of kinase inhibitory activity that extends beyond CDKs 4 and 6. And we know that in the clinic that abemaciclib has a distinct toxicity profile and also, to some extent, a distinct activity profile, and it has a much higher response rate when given as a monotherapy compared to the other drugs. What's interesting, however, is that when we combine these drugs with hormonal therapy in patients, the efficacy of all three agents is very similar. And there is a body of evidence suggesting that their activities in cancer cell lines may also be very similar. So the studies that come to mind, which I think speak to this point, first of all, a large study performed by scientists at Eli Lilly, where they profiled the sensitivity of hundreds of different cancer cell lines to palbociclib and abemaciclib, and found that sensitivity to one drug was very well correlated with sensitivity to the other. So that would suggest that they're both working on a similar target. And then another study published by Eric Knudsen's group, where they showed that although in vitro, abemaciclib and palbociclib induced very different gene expression profiles in tumor cells, in mouse models looking at tumor tissue, the transcriptomic changes were much more similar. So I actually think this is probably one of the biggest questions that everyone in the field discusses on a daily basis, and we still don't have a clear answer. My gut feeling is that the drugs are different, but at least with respect to the phenomena I described today, I think these are CDK4 and 6 related.
Excellent. Well, unfortunately, we have reached our time limit. We have a very long list of questions, which speaks to, a fantastic talk. And Claudia will send you those questions. But again, thank you so much, Shom, for getting up so early or late, however you want to look at it, and presenting your phenomenal work to us today.
Thank you, Sandy. It's absolutely a pleasure. As I say, I'm still, large part of my heart's very much still in Boston, and so it's an honor and a pleasure to be able to present to you all.
Great, thank you.
All right, thank you.